the moat coral nursery down in the lower Florida Keys, and then if the results we found in the nursery were predicative of any of the outplant results that we saw. So for those of you that aren't familiar with the lower Florida Keys, it's off Monroe County, past the 7-mile bridge on your way to Key West. And the stars highlight where the moat nursery is, along with my outplant sites, Site P and Site U, in reference to some of the more familiar reef sites that are down there. And the purple box is the Looe Key existing management area. So as I said, we wanted to see what was the difference between the two production methods that they use, the block method, which is on the left, or the tree in the middle, and then is that predicative of all of what we would see after outplant? So initially I used 4 different randomly selected genotypes out of the nursery, and we replicated between the tree and block method. We also had temperature loggers on the ground and up at the tree height to see if there was a difference at all, and we found that there was not a significant difference in temperature at all, so we just combined them for the temperature data points that you see up there. So this year was in 2015, and we did have a significant bleaching year, so these corals were in a lot of stress for a part of that time. So it was interesting to see that even though during the stress of up here, which 31 degrees is about the highest temperature that these staghorn coral can handle, that we still had a period of high growth here. And we found a lot of interesting things. So there was significant difference between the blocks and the trees, but growth rankings stayed consistent between both of the methods. So our fastest growing genotype on the block was also the fastest on the trees, vice versa. And so we're just going to keep in mind here that 5 was the best as far as the trees, 4 was the worst, and this was same results for the blocks. 5 best, 4 worst, and this will be interesting once we get to the outplanting part. Another part that we did look on here was also if there was a difference in growth on tree levels. For the trees, there's typically 5 levels that the corals are hung on. We did not see any significant difference at all between the heights on the tree as far as growth. So after we outplanted at the 2 different sites, we found that there was a significant difference in the growth again between all of the different genotypes, but there wasn't a site by genotype difference. And then also interestingly, we found that there was not a significant site effect at 6 months, but there was after 12 months monitoring. Now we also added an extra genotype into our outplant study that was known as a super nursery genotype because it was able to withstand bleaching, disease events, and was one of their fastest growers in the nursery. So we wanted to see if that also translated after it was outplanted. So what we found was that results of growth kind of flip-flopped from what we saw in the nursery. Our slowest grower in the nursery, number 4, was almost one of the fastest growers, and 54, which was one of the medium growers, was the fastest, and 5 was no longer the fastest anymore. And these 2 were both significantly different in the nursery, but were not significantly different after outplant. And you can see how growth changed over time. While it was still significantly different between genotypes, growth seemed to stabilize between all of the different genotypes, with only one being significantly different from the others. And our super genotype was over here, number 7. So he didn't really do much better or worse than the other genotypes, which was interesting to see because one of the things that coral nursery managers are wanting to look at is if you can just start outplanting those great survivors from the nursery and think that they're going to survive just as well after outplant. And this is showing that it's not looking like that is going to happen. So what we found that, yes, growth rankings seem to stay consistent across the different grow out methods that are used within moat, so a benthic and a suspended method. And there is difference in performance within the genotypes, which we've heard from some other talks that are also found. And the nursery does not seem to be predicative of your initial outplant, so it may be better to start still with, or continue on with outplanting a diversity of genotypes versus your better producers within the nursery. So I only just squeezed up my little contact information up here, so if anybody else has any questions, I'll meet with you at the after party.